Hello everybody. Now we have come to the third lecture of the course. In this lecture, in the next half an hour or so, we will discuss very important characteristics of fruits and vegetables, that is respiration, ripening and senescence. The broad concepts that I will cover include respiration and its significance, how can we measure respiration rates, or what are the various factors which influence the rate of respiration. Then we will also discuss ripening and senescence, and finally transpiration and condensation, which is another very important factors which influences the quality of fruits and vegetables. So, let us start. Let us see what does respiration means. It is the metabolic process by which cells convert matter into energy, which primary constitutes of enzymatic oxidation of organic substrates in the presence of oxygen. You can see in the figure, it is shown an apple, which inside it contains sugars. We have indicated glucose. Then after harvest, these fruits or similarly other vegetables, they consume oxygen, where these sugars are oxidized, and finally as a result of respiration they produce carbon dioxide, water and energy is generated. Okay. The respiration may be of two types, aerobic respiration and anaerobic respiration. Aerobic respiration consists oxidative breakdown of organic substrate such as carbohydrate, protein, lipids, etc. In the anaerobic respiration, limited oxygen causes fermentation of glucose, leading to the formation of alcohol, and in the process various metabolic pathways do their jobs, which I am not going to discuss here. That is the metabolic pathways, glycolysis, tricarboxylic acid cycle, electron transport system, etc. But I recommend you all that as you must have that is some knowledge of this, please read from any biochemistry or plant physiology books, you will get the details of it. Here we will basically consider the science and engineering aspects of these things. So, what is the significance of respiration as far as the post harvest processing and physiology or handling transportation of fruits and vegetables are concerned? In the respiration, there is a loss of substrate, that is utilization of substrate in respiration causes loss of food reserves in the tissue, there is loss of taste and other quality attributes, and also there is loss in weight during respiration. As I told you in the earlier slide, in the respiration there is a consumption of oxygen. It is important in maintaining aerobic respiration. The reduction in oxygen concentration below 10% controls the respiration rate. That is, it slows down the senescence. That is, if you lower down oxygen concentration in the gaseous environment where the material is kept, the rate of the respiration will go down, and obviously the rate of senescence process will be slowed down. Exactly opposite of this is the carbon dioxide production, that is in the respiration carbon dioxide production it increases, there is accumulation of carbon dioxide, and this can be beneficial or harmful depending upon the commodity's tolerance to elevated carbon dioxide. Effective measures for delaying senescence in control atmosphere storage or modified atmosphere packaging. In fact, whole principle of the operation of the CA storage or MA packaging is based on the respiration, that is providing the O2 as a slowest possible rate and maintaining the carbon dioxide concentration little elevated depending upon the commodity, and the respiration is allowed to continue by maintaining proper O2 and CO2 in the environment. We maintain slowest possible respiration. Details of course all this CMA we will take up in some class later, but mind it, the material is in good condition as long as it is respiring aerobically. Once aerobic respiration stops, anaerobic respiration will take over and the material will spoil. Release of heat. The heat generated in respiration rate raises the temperature of the produce. Respiration rate helps in designing the packaging system, storage facility and calculation of refrigeration load during storage and transportation. Respiration rate or respiration behavior of a commodity is very good indicator of its shelf life. Respiration determines the post harvest physiology and deteriorative ability of the produce. Respiration rate indicates the storage potential of the plant produce. Higher the rate of respiration, shorter will be the shelf life of the produce. Then quality changes. Controlled respiration enhances the quality like color development, softening, astringency, loss, aroma, etc. Extremes in respiration rate results in development of specific physiological disorders resulting in loss of quality. Respiration is beneficial in providing carbon skeleton intermediates for pigment synthesis, flavor development, formation of ripening enzymes, fat, sterols, etc. Respiration behavior, rate of the respiration is also considered an important criteria to compare perishability of the fruits or vegetables. Here I have shown like respiration rate, it can be measured either in the terms of oxygen consumed or in terms of carbon dioxide produced. So, if you see in the terms of carbon dioxide produced, that is milligram carbon dioxide per kg per hour. So, at a temperature 10 degrees Celsius, the carbon dioxide produced is less than 10 milligram per kg per hour, or at 20 degrees Celsius if it is less than 40 milligram per kg per hour, the produce is categorized as a very low respiring, that is it will have a good shelf life at room temperature. Examples of this category include nuts, dates, dried fruits, etc. The other similarly it is indicated shown in this table, you can see like for example, if the carbon dioxide production is in the range of 10 to 20 at 10 degrees Celsius or in the range of 40 to 80 at 20 degrees Celsius, the commodity is considered as moderate respiring. The examples of this pepper, carrot, tomato, citrus fruits, banana, etc. High respiring commodities, that is those who evolve, produce carbon dioxide at 10 degrees Celsius in the range of 20 to 40 milligram per kg per hour, or at 20 degrees Celsius 80 to 120, they are highly respiring commodities. Examples are pea, radish, apricot, and very high where the carbon dioxide production is more than 40 at 10 degrees Celsius or more than 120 milligram CO2 per kg per hour. Then the examples of this highly respiring fruit or vegetable include mushrooms, green onion, the cauliflower, blackberry, raspberry, and so on. So, this is here rate of respiration is very important, and this once you understand this, if you know that whether accordingly the post harvest management can be done. Okay. Now, and the important that how to measure the respiration rate. The rate of respiration of the fresh produce can be, as I told you, measured using gas exchange, either the O2 oxygen consumption rate or CO2 production rate. So, there are three experimental methods which are generally used. This is closed system method, flushed system method, and permeable system method. So, in the closed system method, as you can see here in the figure, that a gas tight container of known volume is filled with the commodity. Okay. It filled with the fruit or vegetable, and the container which has ambient air in it as a initial atmosphere is closed. So, you keep the material inside the container and the container is closed, and then you can see in the figure that is there are syringes, or it can be connected with the headspace gas analyzers. So, the changes in the concentration of O2 and CO2 over a certain period of time are measured and used to estimate the respiration rate. Respiration rate can be, as I told you, measured either in the terms of oxygen consumption or in terms of carbon dioxide production. In terms of oxygen consumption, that is the concentration of gas concentration volume by volume at a particular time, and the same concentration may be oxygen or carbon dioxide at a time t plus 1 divided by 100 multiplied by time difference delta t, whole thing multiplied by Bf or W. So, this either, that is you can say RO2 or RCO2 is the rate of respiration, that is either cubic meter O2 or CO2 per kg per second. YO2 and ZCO2 are the gas concentrations of O2 and CO2. T is the storage time in seconds. Delta T is the time difference between two gas measurements. Okay. Bf is the volume of the respiration chamber in cubic meter, and W is the weight of the fruit that has been, or weight of the commodity that has been kept in the chamber. So, in this way you can find out rate of the respiration. In the second method, flow through system method, the product is enclosed in an impermeable container through which gas mixture flows at a constant rate. As you can see here in this figure, the material is kept in a impermeable container, and there is a facility for gas inlet and gas out. The respiration rates are calculated from the absolute differences in the concentrations between the outlet and the inlet when the system reaches steady state. Okay. Again, here also RO2, either you can consume RO2 or you can calculate RCO2, that is RO2, YO2. YO2 is the concentration of oxygen, that is inlet, minus YO2, that is outlet. Inlet concentration minus outlet concentration of oxygen divided by 100 multiplied by, that is the flow rate of the gas in cubic meter per second, and divided by weight of the fruit in kg. So, that is the, similarly you can calculate the carbon dioxide production rate in this method. Then the third method, permeable system method. Here a package of known dimensions and film permeability is filled with the product. So, that is, it is packaged with certain films, and the permeability of the film, that is oxygen permeation rate or carbon dioxide permeation rate, is known. The steady state concentration of O2 and CO2 are determined, and a mass balance is performed on the system in order to estimate the respiration rate, and RO2 or RCO2 can be calculated, that is the RO2 is equal to PO2, that is the it is the permeability, oxygen permeability of the film multiplied by area divided by 100 multiplied by L is the thickness of the packaging material in meter, and M is the weight of the produce. So, whole thing multiplied by, that is external oxygen YeO2, that is external oxygen minus internal oxygen concentration in the chamber. So, that through this you can calculate RO2. Similarly, the other equation which is given here O2 is replaced by CO2, that other attributes remain the same. So, one can calculate the respiration rate on the basis of the either oxygen in or CO2 out, produce CO2. So, if we have in this table, I have compared that these three methods, like if you see that a closed, all these three, closed method, flow through or permeable method, they are all three are non-destructive, that is the samples that they are not destroyed, that is the sample remain as such. All these three are time and labor consuming. If you look compare this from the simplicity or complex, that is the closed system is simple, whether the flows through and permeable system are little complex. Ability to test different combinations of gases, yes, that is closed system and flow through system are good, but in the permeable system it becomes difficult. Suitable for low respiring products, that is yes, closed system, and permeable system is suitable for low respiring products. Okay. That is the flow through system and permeable system are suitable for high respiring products, and that is the accuracy, that is what is the major factor which will influence the accuracy of the RO2 or RCO2 measure. So, it is the free volume in case of closed chamber, that is in the flow through system it is the flow rate of the gases, and in the permeable system the permeability of the packaging material, package dimension, steady state concentration, etc. These how accurately these are maintained, how accurately these are measured, on the basis of this you will get the accurate value of the rate of the respiration. Then another very important characteristics is the RQ, that is called respiration quotient. The respiration quotient is the ratio of the CO2 produced to oxygen consumed. RQ you can see in the figure it is equal to RCO2 divided by RO2, and the value, if you get the value of RQ as 1, it means that the respiring material or oxidizing material is carbohydrate. Your total oxidation of 1 mole of hexose consumes 6 moles of oxygen and produces 6 moles of carbon dioxide. If it is in the case of organic acid, the RQ will be more than 1. In the case of lipids and protein, if they are respiring oxidizing, the RQ will be less than 1. When there is a fermentation reaction, that is anaerobic respiration, RQ will be more than 1.3, means that is production of ethanol is resulting in decarboxylation of pyruvate to carbon dioxide without any uptake of oxygen. Okay. In the organic acid, when it is more than 1, the presence of inherent oxygen molecule in the acid, organic acid, that can oxidize, and it decreases the requirement of the external O2. So, these are the RQ value obtained is a good indicator of the substrate which is getting oxidized in the respiration process. As far as the factors affecting the rate of respiration, obviously I told you that different factors like they affect the rate of respiration. Among this, temperature is the one most important. Within the physiological temperature range, the velocity of a biological reaction increases 2 to 3 folds for every 10 degrees Celsius rise in temperature. The rate of reaction rate at 2 dissimilar temperature is called temperature quotient, that is Q10 is equal to R2 by R1 to the power 10 by T2 minus T1. So, by this, that is where R1 and R2 are the respiration rates at temperature T1 and T2. You can find out the respiration rate at 2 different temperature and find out yes by what is the Q10 value, means that is by changing the temperature by 10 degree, how much the rate of respiration will increase or decrease. Okay. And that will provide you important input to have the to manage the commodity post harvest during its handling, transportation, etc. Similarly, the concentration of oxygen, if O2 concentration is reduced below 10%, a significant drop in the respiration rate is observed. If the O2 concentration is below 2%, it will initiate anaerobic respiration in many commodities. In the figure, you can see the effect of O2 concentration on respiration, and as is clearly indicated by the figure, in the figure, that super atmospheric O2 concentration, when it goes more than 20.9%, it slightly stimulates respiration rates. Concentration of CO2, again elevated CO2 level reduces aerobic respiration. At 20% carbon dioxide concentration, anaerobic respiration significantly increases. There is a ethanol and acetaldehyde accumulation. Carbon dioxide induced physiological disorders result in reversible tissue damage. Okay. The figure shows here that effect of CO2 concentration on the rate of respiration. CO2 concentration increases, the rate of respiration decreases. Okay. The stress, like physical stress such as impact bruising stimulates the respiration rate of fresh vegetables. Any mechanical injury such as cutting, abrading, slicing and shredding increases the respiration rate. Water stress, which is induced by the lower than optimum relative humidity in the air surrounding the commodity, can stimulate the rate of respiration. Biological stress such as incidence of disease also causes increase in the respiration rate. Other stresses that stimulate the respiration rate of vegetable include exposure to ionizing radiation and various chemicals such as methyl bromide and other fumigants, etc. In this figure, just I have tried to show you that how the rate of respiration is related to the shelf life. Lower the respiration rate, like you can say in the potato, it has the highest shelf life. At room temperature, we are talking about highest respiring commodities, that is like mushroom, they have the lowest shelf life. So, that is one in the post harvest handling and management. This respiration, you have to manage the respiration at the slowest rate by manipulating the environmental conditions, O2, CO2 composition and other factors, which we will take up separately when we study this aspect. Okay. So, after respiration is done, now let us talk about ripening and senescence. In some of the fruits, etc., material, respiration is directly related to ripening as well as senescence, particularly in the case of climatic fruits. Ripening refers to physiological and biochemical changes of a fruit to attain desirable color, flavor, aroma, sweetness, texture and thus eating quality. Once the fruit, you see the mango or such other fruit, mango, green mango, how tart it is, sour it is, but once it ripes, becomes yellow or other color, it becomes very sweet. There are many fruits. So, lot of changes, physical changes, other changes, many sugars are produced, starch is converted, that we will take it little later in the next slide, but this fruit becomes good, its quality, eating quality becomes good. The process of ripening usually does not occur until a fruit reaches its full maturity. Ripening of a fruit may occur on the plant or after harvest, depending upon the species. Some species they ripe on the plant itself, some species even they ripe after they are harvested. Ripening in fruits follows the physiological maturity and precedes senescence, which leads to the death of the tissue. Okay. Senescence, that is when a fruit has its or passes its maximum ripeness. Once the climatic rise, you will see that it comes to the peak of the climatic, that is the maximum stage good eating quality, and after that there is a decrease in the rate. Okay. So, means that is when the fruit has reached maximum ripeness, when it passes the maximum ripeness, it begins to break down and decay. Rather than a simple breakdown process, senescence is the final phase in which series of normally irreversible physiological and biochemical events is initiated, which leads to the cell breakdown and death of the fruit. And you can see here in this figure also, that is initially it is the ripening stage, fruit ripening and development stage, finally it has come on its maximum maturity or ripening, maximum quality, and then senescence, that is in the strawberry you can see in the picture. Okay. The ripening and senescence, that is in fact you can see in the picture here what I already discussed, elaborated, that is there are three stages: growth, ripening and senescence, and it is shown here that is the growth stage. Okay, in the both in the climatic and non-climatic fruits, that is in the climatic fruits at the end of the organoleptic growth stage, the respiration increases. That increase in the respiration is related to, there is also increase in the associated with the increase in the ethylene production, and it reaches maximum, that is here it reaches maximum, that is the climatic maxima, peak has been obtained, and after this it goes down. Okay. So, in the organoleptic ripening, that is optimal flavor and aroma reached and the produce is suitable for consumption, we say. In the physiological ripening, that is optimal size is to be harvested, it shows. So, in the non-climatic fruits, the respiration rate decreases gradually without the ethylene production. So, that is the ripening and senescence, how they are related. Here, this in the I have shown the climatic fruit ripening. Again, I expressed that in the climatic fruit, the patterns of ethylene production and carbon dioxide increase, rate of respiration increase in the climatic fruit. That is, ethylene is a ripening hormone. So, once that is initiated, after the fruit is harvested, there will be a slight decrease in the rate of respiration. It will come to the climatic minimum, that is ethylene production. Then here, when it comes to climatic minima, both there is an increase in the ethylene production as well as in the increase of respiration rate. It comes the finally climatic minima and then comes to the senescence stage. So, that you can see here in the means horticultural produces can be divided to that is the climatic start of ripening is accompanied by a rapid rise in the rate of respiration called respiratory climatic, and after climatic the respiration slows down as the fruit ripens and develops good eating quality. The examples of these climatic include banana, apple, melon, papaya, mango and tomato. Non-climatic, there is a after harvest there is a continuous decrease both in the carbon dioxide production, that is rate of respiration, as well as ethylene production, there is continuous decrease. So, eating quality suffers if harvested, the commodity is harvested before they are fully ripe, because their sugar and acid content does not increase. Okay. The respiration rate slows gradually during growth and after harvest, and maturation and ripening are the gradual processes in the case of non-climatic fruit ripening. Examples of this category include cherry, grape, lemon and pineapple. So, what are the various chemical and physical changes that occur during ripening? Like color, there is a pigment, there is a loss of chlorophyll, green color converts into various yellow color, red color, other colors, synthesis of carotenoids, there is synthesis of anthocyanins, etc. The color improves. Similarly, texture, it becomes soft. It is because there are changes in the pectin composition, alteration in the structure components of the cell wall, there is a hydrolysis of storage materials, etc. Various other carbohydrate composite stages, organic acid, various aroma, volatile compounds are formed, which improves the results in the improvement of the flavor during this ripening. Also, the inner respiration, that is the sudden peak in the respiration for climatic and gradual decline in the non-climatic fruit. Ethylene metabolism, that is sudden peak in the production of climatic food and constant production of ethylene in the non-climatic fruits, etc. And it increase in the tissue sensitivity due to ethylene. So, that is in fact, so that is the during climatic period in the ripening food, there are a lot of changes that take place. Okay. So, after the respiration, now let us see another very important characteristic, that is transpiration. Transpiration is a type of water evaporation or water loss from the plant parts. Transpiration involves the evaporation of water from cell surface into intercellular spaces and the diffusion of water molecules out of the plant tissues or organ into the surrounding air. The water loss from the fruit and the vegetable induces water stress in their tissue, which may enhance or accelerate senescence in commodities due to increased rate of cellular membrane disintegration and leakage of the solutes. And you can see here these three, that is bell pepper, fruit immediately after harvest, the left in the middle 3 days after harvest, and the right 7 days after harvest. The fruit is kept at 20 degrees Celsius and 70% relative humidity, and you can see how means that it is transpiring, moisture is losing, and this loss of the moisture is related to it, leads to transpiration, leads to loss in weight, loss in appearance, loss in textural quality like softening, flaccidity, limpness, crispness and juiciness, etc. various. So, even loss in nutritional quality, you can see then the tomato, it loses glossy appearance in products. This is grapes, that is all these are the effect of the transpiration. It is the fresh pomegranate, B is the which is loss of appearance, R which is the loss of shrivelance, that is shriveled, there is a significant amount of transpiration has taken place. So, you see this is transpiration is very because the moisture is losing. So, this critical moisture loss upon which the produce becomes, it loses its market value, that is when you go into the market, that is for example cauliflower, if it loses its moisture content due to transpiration up to 6 to 8%, or celery 8 to 10%, it will lose its market value. Beans 5 to 7%, asparagus 7 to 9%, sweet corn 5 to 10%, tomato 6 to 8%, or lettuce 3 to 5%, onion 8 to 12%. So, these are the critical moisture content, means if that is why it becomes very important that this transpiration, okay, one should have to manage in post harvest management and make sure that they do not lose the moisture, otherwise they will lose, the product will lose its market value. Okay. All right. So, this is the vapor pressure deficit, where vapor pressure of water, the vapor pressure of water is the equilibrium pressure of water vapor above liquid water or solid water. It is the pressure of the water vapor molecule resulting from evaporation, and relative humidity is the water content of the air expressed as percentage relative humidity to the percentage relative to vapor pressure of the same air saturated and at the air at the same temperature. So, P air, that is the partial pressure of air at a given temperature, is equal to the partial pressure of the saturated air at the same temperature multiplied by relative humidity, and this vapor pressure deficit delta P is equal to P P, that is the partial vapor pressure of the water at surface of the produce minus P air. Okay. And with this you can calculate the rate of transpiration, like TR, rate of transpiration in kg per second is equal to K AS delta P, that is the K is the overall mass transfer coefficient, AS is the surface of the product, and it is also equal to delta P by 1 by K AS, or finally 1 by RE. RE is the resistance to transpiration. So, you can see that rate of transpiration is directly related to the vapor pressure deficit, or it is indirectly inversely TR is proportional to 1 by RE. So, how can we, that is what are the factors that is affect the rate of transpiration, like skin structure, size, shape and surface area, vapor pressure difference, air movement, heat of respiration, etc. All these all these commodities influence the rate of respiration. Okay. Like for example, high vapor pressure difference increases the water loss, higher ratio of the surface to volume, the greater the loss of the water by evaporation. Air circulation increases the moisture evaporation from the surface of the produce, increasing the water loss. Okay. Similarly, the level of maturity of the commodity, immature fruits tend to encounter higher transpiration rate than the mature fruit, and this is particularly due to the permeability of the skin to the water vapor. Amount of the solutes present in the produce, since the solute depresses the water activity of the solution, higher solute concentration in the tissue binds the water and it reduces the water loss, and ultimately temperature and relative humidity, high surface temperature and low relative humidity increase rate of the transpiration. So, what are the remedies? How can we minimize the transpiration? That has to be done in order to have a better marketability, better shelf life of the produce. So, the water content of the environment should be kept as close as possible to the water content of the product, maintaining low temperature and high humidity in the storage, protect the product from mechanical and physical injuries, application of waxes and other resisting coatings, water resisting coatings on the surface of the produce, selecting suitable packaging material and packing appropriately, or equilibrium humidity packaging, like maintain the equilibrium inside the packet and outside the environment is proper, so that there is no movement. Then another very important characteristics is the condensation, water when it transpires, if the packaging is not proper, then there will be water condensation. It refers to the conversion of water vapor into liquid water. It happens when the temperature drops below the dew point and relative humidity reaches to saturation. The condensation may be on the surface of the produce, as you can see here in the picture, or it may be on the packaging film. Okay. The condensation at the surface of the produce, okay, it leads to the microbial growth, like free water on the fruits and vegetables will enhance the microbial growth, leading to decay and loss of produce. Free water promotes germination of the fungal spores and pathogens. This and the condensation and the packaging film affect the film permeability and lead to aerobic respiration. Okay. So, the condensation on the surface of the produce, when the surface temperature of the produce falls below the dew point temperature of the surrounding air, the water vapor in the moist air precipitates as droplets or forms a thin layer of the surface of the solid. Condensation occurs due to temperature fluctuations under high relative humidity in the supply chain and also during rewarming of the cold fruits. Condensation at the packaging material, that is, whenever the packaging material has lower water vapor transmission rate than the product transpiration rate, water molecule evaporated from the product will remain in the package, enhancing the water vapor pressure inside the package and leading to the water condensation. So, these are the very basic principles which lead to, so one has to, in the packaging, one has to take care of all these things, like remedies for minimizing the condensation, storing the produce at a very stable temperatures, make sure that there is no fluctuation of the temperature, maintaining a continuous cold chain, temperature conditioning of packing material, especially plastic bags, so that they are at the same temperature as the produce prior to packaging, and as far as possible packaging in a cold room, which usually always is not practical. Then again here also equilibrium humidity packaging will be beneficial. Cooling decay sensitive produce to above the dew point temperature until they are packed, and then cooling to the desired storage temperature. Faster warming of the cold fruit can reduce the length of the time of the produce is wet and therefore decrease the condition for disease development. With this, I will now summarize this lecture, that respiration rate is an indicator of the shelf life of fruits and vegetables. Temperature, oxygen, carbon dioxide, gas concentrations are the main factors affecting the rate of respiration. Ripening of climatic food is indicated by increase in the CO2 and ethylene concentration, while it slows down in non-climatic fruits. High transpiration rate causes loss in weight, appearance, texture, nutritional value of the product, leading to the loss of the market value. Water condensation causes issues of microbial growth of the product surfaces as well as affect the packaging film permeability. Respiration rate, ripening, transpiration rate and condensation rates can be controlled by proper manipulation of temperature, relative humidity, gaseous concentration, gaseous composition, I mean that is environment where the material is kept, like O2, CO2, ethylene, and the stresses on the commodity. These are the references which were used in this. Okay. Thank you very much.